Fellow Stonkers, I am pleased to introduce you to enzotitics. Before embarking on this journey, I have to lay waste to short-term holders. If you get off on get-rich-quick schemes, then this stock is not for you, and you should close this page. Now that we have separated the wheat from the chaff, let's begin. Enzotitics Incorporated is a drug development company committed to the commercialization of its proprietary proteins for the treatment of debilitating infectious diseases. The company's products consist of two distinct drug development proprietary technologies, immunotherapy, and fully human monoclonal antibodies. What are monoclonal antibodies, you may ask? A monoclonal antibody is an antibody made by cloning a unique white blood cell. Monoclonal antibodies can have monovalent affinity, binding only to the same epitope, the part of an antigen that is recognized by the antibody. Molecules from invading organisms, such as viruses, bacteria and other harmful organisms, are called antigens and prompt the body's B cells to make antibodies by the billions. Each antibody has two antigen binding sites, one on each arm of the Y, and works to neutralize these invaders. Effective monoclonal antibodies will be those that target conserved and expectedly immutable virus sites, whether the therapeutics is a monoclonal antibody or a preventive vaccine. Targeting these conserved sites, conserved meaning located across all strains, allows the production of a therapeutic that will not be rendered ineffective due to mutation, variants, of the virus. In other words, even a variant form of the virus will expectedly contain the immutable targeted sites. Targeting immutable sites avoids the ineffectiveness that is experienced when a therapeutics or vaccine targets a site that is mutatable. This exact reason is why Eli Lilly's and AstraZeneca's anti-coronavirus monoclonal antibodies were earlier revoked, due to their ineffectiveness. Prior government funding for producing anti-HIV monoclonal antibodies have failed due to what the industry calls virus escape. The anti-HIV monoclonal antibodies VRC01 and VRC02, produced by Vaccine Research Center in conjunction with the NIH, were tested for years and then abandoned due to the HIV virus mutating around the therapeutic. This has also been seen recently with the. As a result of the acquisition of Bioclonetics Immunotherapeutics back in December 1, 2020, enzotitics has become an expert in the creation of human Ig1 neutralizing monoclonal antibodies, not humanized mouse or rat counterparts, as are the majority of monoclonal antibody therapeutics in pharmaceutical use today. Why is this important? When a humanized monoclonal antibody is administered, a condition called HAMA, human anti-mouse antibody, can develop in the patient. HAMA is an antibody found in humans which reacts to immunoglobulins found in mice. The HAMA response is essentially an allergic reaction to the mouse antibodies that can range from a mild form, like a rash, to a more extreme and life-threatening response, such as renal failure. HAMA can also decrease the effectiveness of the treatment, or create a future reaction if the patient is given a subsequent treatment containing mouse antibodies. It has been observed that anywhere from 1/3 to more than half of patients receiving mouse-derived antibodies will develop some form of HAMA response. I assume that if you have a TV, you have been seeing monoclonal antibody therapy frequently talked about on all the major news outlets. Particularly one monoclonal antibody cocktail, RegCov2, has received significant media attention since August 21, 2021, the day Florida Governor Ron DeSantis announced that the state of Florida would open a new monoclonal antibody therapy treatment site. This is the same antibody cocktail that former President Trump received when he was infected with COVID. The enzotitics process differs greatly from the process used by Regeneron. Regeneron antibodies are produced by the company's VelocImmune mice, which have been genetically modified to have a human immune system. Enzotitics process does not use mice with a genetically modified human immune system. A primary distinction of the enzotitics process for creating fully human monoclonals is that production begins from taking immune B cells from humans who have survived successfully from a natural coronavirus infection. These antibodies will retain the original natural antibody affinity and specificity and have a lower risk of immunogenicity when used as a therapeutic. They are expected to provide broad-spectrum coverage against viral variants with increased potency, stability as a single-domain molecule, and, in the recombinant form, will have accessibility to the virus epitopes, binding sites, not accessible with a whole antibody. The company's process for producing monoclonal antibodies against infectious diseases begins with conducting an artificial intelligence (AI) analysis of the epitope sequences to identify conserved sites with a conservativeness of up to 99%. Conservativeness meaning the percentage of identical or similar sequences in nucleic acids or proteins across species of virus. In the case of the coronavirus, The company initially analyzed 50,512 coronavirus isolates and has now analyzed over 1 million isolates, resulting in the identification of 19 conserved, immutable sites. This was the largest known repository of isolates ever screened. These 19 conserved sites were confirmed as 98.71% to 99.29% conserved over the entirety of all 1 million coronavirus isolates analyzed. From further analysis of the Delta variant, Lambda variant, Alpha, Beta, and Gamma variants, the company has confirmed that the 19 sites against which it is producing monoclonal antibodies exist in all of these variants. Although Enzolitics has long recognized the immense value of monoclonal antibodies, in order to be effective, monoclonal antibodies produced against viruses must target immutable, conserved epitopes, sequences, on the virus, or the antibodies will be ineffective due to virus escape. For example, during the first few weeks of infection with HIV, the human body mounts an immune response. But, as with treatment failure on antiretrovirals, mutated viruses that escape the immune response quickly thrive and replicate to become the predominant viral population in the body. In the language of microbiology, CD8 cells become unable to recognize the mutated epitopes of these mutated molecules, and the therapeutic quickly loses efficacy. This is precisely what occurred with regards to the anti-coronavirus monoclonal antibodies recently produced by Eli Lilly and AstraZeneca, antibodies now withdrawn from the market.